Yeah, good morning. Welcome to everyone that's uh, joined in to attend our last session of Regenerative Ag Week. Want to remind you that you can ask questions through the Q&A button and the chat feature at the bottom of the screen. Please don't be afraid to put those questions in there as you think of them. We'll hold those questions to the end, and then we'll go through them in a manner how they came in. And so go ahead and do that. This is the last week 8, day 3. So for those of you that kind of stuck with us throughout the sessions, thank you. Appreciate that. And today we have Dr. Vander Nunes. He is relatively new to SDSU. He's our sustainability research and extension. I believe you have both appointments, Vander, and so
Right.
he comes from Illinois, working under a great scientist there, and I'm sure he'll maybe mention that a couple times. So um looking forward to his presentation today. So take it away, Vander.
Well, thank you very much for your introduction, Anthony. And thanks everyone for being here today. I'm going to be kind of looking to the side because I'm using a bigger monitor here. It's a pleasure being here today. You're going to notice I have a different accent. I'm from Brazil. And today let me go ahead and share my screen here. Right. I was talking to Anthony and Clarence and Matthew that I just finished preparing this presentation. I haven't presented about this topic before, and I decided to bring this topic to talk about it with you today because I think it's extremely important topic when we think about regenerative agriculture, soil health, sustainable ag, and it's something that I did not learn during undergrad and even during grad school. It's something I just learned later on in my career from my former advisor. This presentation is going to be mostly focused on two of his publications, and I talked to him, and he said, "Sure, spread the word," and I hope you guys everyone in the audience can take some can have some of these messages to take home and use it or not according to your preference. So here's just the outline, everything that I'm going to be touching a little bit about today, and every single slide is numbered here. You probably can see my mouse. Feel free to record the number of the slide if you have a question, write it down, and I'd be glad to take questions in the end. To start with, since it's a regenerative agriculture week, I decided to bring these two guys here, and specifically these two guys, these two scientists, because one is in Illinois, did his most of his career in Illinois, and this is Cyril Hopkins. He's actually from Minnesota. He was born in the border of Minnesota and South Dakota. He did live in South Dakota for a while, and ended up as a professor at the University of Illinois and a scientist there. And there, for years, he created, after years of research, he created this publication where he entitled as the Illinois System of Permanent Soil Fertility. And this is 1913. We didn't even have the use of synthetic or commercial fertilizers back then. Fertilization was basically when we talk about inputs in ag, we would be more talking about manure, rock phosphate, and limestone. That's about it, and some other organic forms. The synthetic fertilizers we see nowadays, MAP, DEP, urea, UAN, they didn't exist back then. They had just been discovered, the process to produce them. And look at the principles of his system: nutrient management, diversified crop rotation, cover crops, grain and livestock manure, integrated farming. And then Joseph Hutton, he is a scientist, did his career here at SDSU. Uh, he went to Illinois for a presentation. He studied there too, and there he met Cyril Hopkins, and they kind of worked in the same line. Joseph started to work following Hopkins' research line and just added on top of what he had seen as a sustainable or regen ag. How can I say? They didn't use these specifically terms back then. He added practices such as windbreaks and leaving crop residue after harvest. And here are the pubs. I definitely recommend taking a look. These are long. They're like each of them are like 40, 100 pages. They're long, but I definitely recommend taking a look. Good stuff. And when we bring it to today, right, to 2025, I just took a print screen from the Soil Health South Dakota Soil Health Coalition, and the principles are basically the same. It hasn't changed much. The only one that it's worth mentioning is limited disturbance. It's important to remember that back in the 1913 30s, we didn't the no-till practices weren't yet used in the US. Is something that, if I'm not mistaken, started in the 60s. And we also didn't have herbicides to control weeds. So it's something that just appeared later on. So this is one of the reasons why they didn't mention it in their practices. But other than that, it's pretty much the same. And today we're going to be putting a lot of focus on nutrient management, mostly on nitrogen. So here is the nitrogen, and it's quite interesting because there is a lot of nitrogen in the. We're surrounded by nitrogen, uh, and yet we still have to apply nitrogen. Look at this soil here, for example. This is a soil profile in Aurora, where the students learned introductory soil course. It's just a soil pit, and just on the surface of this soil, I just made some assumptions here, but they're fair assumptions. And just on the surface, you would have about 4,000 pounds of nitrogen in 1 acre. And on the atmosphere, you have 78% of the nitrogen gas. That would be around 33 US tons per acre. So we're surrounded by nitrogen, but unfortunately, most of this nitrogen cannot be taken up by plants. They are present in either inorganic forms that cannot be used or organic forms that need to be mineralized before being used by plants. And we're going to talk a little bit about these today. So this is the global demand. Why nitrogen, right? There are so many different many reasons for nitrogen. Nitrogen is used in so many, well, to make it simple, nitrogen is used to produce proteins, and all enzymes are proteins, and enzymes are used by all organisms for all sorts of different, to accelerate and or facilitate reactions. So and when you look at the quantities, it's the macronutrient used the most, those that we supply via soil, in terms of via fertilizer, actually. Of course, not more than carbon, oxygen, and hydrogen, but those, we don't count them because we provide them via water and photosynthesis. But it basically doubles what the demand for phosphorus and potassium, for example. The problem is, this is a nitrogen molecule, and they have these three covalent bond. How do we provide nitrogen in agriculture, right? So how do we take nitrogen out there and bring it in? Well, there's basically three different ways to bring in new nitrogen or break down this nitrogen and transform this in a more reactive form of nitrogen that can be used by plants. And these three different processes would be biological nitrogen fixation, chemical fixation, and atmospheric deposition. We're going to briefly talk about these two. And the atmospheric deposition, it's something that happens in smaller scale. We're talking about, it varies from area to area, but less than 20 pounds per acre per year is a fair estimate, according to the literature. So BNF, when we talk about biological nitrogen fixation, to break down the triple bond takes a lot of energy, lots of energy. To be precise, organisms need 18 ATPs to convert one ammonia, one nitrogen ammonia, one into one nitrogen in one ammonia, in two ammonias in this case, right? So it's a lot of energy. If you make some calculations, assuming aerobic respiration, these would be about 3 carbon molecules per nitrogen fixed. And for this very reason, you're going to see the symbiotic organisms, such as the relationship between soybean and rhizobia, as a very successful example, because in this case, you have a plant host that provides everything these organisms need. They have a house. In the absence of oxygen denaturates the nitrogenase, which is the enzyme needed for the process. And the house, the plants, to supply all the carbon needed in the form of photoassimilates. So these organisms can fix as much nitrogen as they can in an exchange with the plant. On the other hand, you have this so-called free-living nitrogen fixers. And nowadays, you're going to see a lot of biological products out there that they're going to say you can apply it either directly to the soil, or sometimes you coat a seed, and that stuff's gonna provide nitrogen, and therefore you're not gonna have to apply as much synthetic nitrogen fertilizer to your soil. Well, one thing I don't want to go deep into this topic, but one thing I can talk to you is the free living organisms. They're very diverse. They can be heterotrophs. They can either they can be autotrophs, aerobes, anaerobes, and they need to find their own way to fight for their own success out there. They rely a lot on soil conditions in order to be successful. That's what I had to say on this slide, and let's move forward, otherwise I'm going to spend too much time here. Another way to fix nitrogen is the chemical fixation, and this is Fritz Haber. He's a German chemist. In 19, if I'm not mistaken, 5 years before here, uh, uh, he won the Nobel Prize that he won because he discovered the chemical process to convert the nitrogen or nitrogen gas in ammonia. And in essence, it's a process that takes a lot of energy. There's a Bosch. You probably have heard the Haber-Bosch process, about the Haber-Bosch process before. The big name here is it's Haber. Bosch just helped Haber to industrialize the process so you could produce it in a large scale. And the main takeout here is you need a lot of energy. You can do it anywhere. It's unlike phosphorus that you need a mine. You need to be lucky to have the natural resource available in your country. Let's say this way, such as Morocco, for example, and then go and mine it. For nitrogen, it's out there in the air. All you need is a fuel fossil, such as natural gas, that you can burn to generate high temperatures and high pressure. And therefore, you can generate hydrogen gas either via water or methane and produce your liquid ammonia. That's simple. And one thing that I want you to know is that liquid ammonia produced, also called anhydrous ammonia, anhydrous means that there's no water there, free of water. It's the mother of all synthetic nitrogen fertilizers and other fertilizers too. So in green here, you have the 3 most used nitrogen fertilizers in the US, being urea the most used, and not only in the US, but in the whole world. And as you can see, the percentage, the nitrogen content decreases. And as the nitrogen content decreases, the price usually increases per unit of nitrogen from these sources. And the reason is very simple. You need more. There's more logistic involved to hold it around, and there's more costs involved to produce these different forms. And hydrous ammonia can also be used to produce phosphorus fertilizers, like the phosphate rocks, or when you just mine off phosphorus, for example. They're usually very low in solubility. They're calcium based, so you can react this phosphorus in acid, and then you form phosphoric acid, and in the end, you can react this with anhydrous ammonia to produce the MAP, the ammonium phosphate, or diammonium phosphate. They're both used as P fertilizers, although they have significant amounts of nitrogen. And some other names here, I just mentioned ammonium sulfate and calcium nitrate. Those sources are not commonly used here in the US, to my knowledge, but they're very used elsewhere, very much used elsewhere. For example, ammonium sulfate is largely used in Brazilian forest industry, eucalyptus, for example, and calcium nitrate is largely used in Europe, for example. They're just alternatives. And I have in gray here in the center the ammonium nitrate. It's a great fertilizer, great, very good stuff. We use it a lot in experiments. And the reason for this product to not to be used more is it can be explosive. So it's dangerous. And this is why they combine it with water and urea and to produce the UAN solutions in equal parts of urea, ammonium nitrate, and water. You're going to have either your 28 or your 32, according to how much water you add. If you add a little bit more or not. So each source have its own advantages and disadvantages. We're not going to be able to talk in details about all of them right now. And here is the nitrogen cycle. And this slide is going to scare you off, and that's not what I mean. I'm not going to talk about this nitrogen cycle. I prepared my own, a simplified version, and that's what I'm going to use. However, this is a very good nitrogen cycle. It's taken from the Nature and Properties of Soils. Well, it's probably my favorite book when we talk about basic soils. It's a very good book, over 1,000 pages. I definitely recommend reading for those who, not the whole thing, but if you want to learn more about a specific process, it's a way to go. And here's the simplified nitrogen cycle I prepared for you today, just assuming that I'm applying urea on the soil surface. So here's urea. I decided to put the chemical formula here just for this compound so you can see what it is. It's just a carbon with an oxygen. And then you have two amino groups, this NH2 here. And as soon as you apply your urea in the soil, your soil is abundant in urease, which is the enzyme that cuts down these two amino groups. And if you're applying in the surface, if it's too hot, dry, you may have ammonia volatilization, especially in sandy soils, or it can also happen in soils on the moldy soils here in this region of South Dakota as well. If applying to the surface, and if you have bad weather conditions. And the red here, it's bad for the farmer because now I applied, let's say, 150 pounds of N, and I already lost some. So you're wasting money here. A quick way to prevent this is by incorporating urea in the soil during your application, if feasible. It's a way to make sure that you're going to minimize, if not completely reduce ammonia volatilization, unless you're applying this in a sandy soil. And then that urea is going to be converted into ammonium. And ammonium, I like to call it the filet mignon. It's a mineral form or inorganic form of nitrogen. It's the preferred form of nitrogen by both plants and microorganisms. It's what everyone likes to eat, if that makes sense, because you can easily convert it to proteins. So that ammonium, our goal when we apply a lot of fertilizers, for example, when you do a fall application, if you do, or when you do 100% pre-plant, you're relying on that or believing that most of that ammonium will be held at your cation exchange capacity, CEC. This CEC is basically generated this it's a place where you can hold cations such as ammonium, And the sources are soil organic matter and clays and clay minerals. Another pathway for ammonium is to be fixed into 2:1 clay minerals. Now, this process is not very common, not very talked much about, but it can definitely happen. And in some cases, for example, I worked with a soil in North Dakota that had over 300 ppm of clay fixed ammonium. What happens is your ammonium and your potassium, they're the very same size in terms of hydrated radii, and this ammonium can get trapped. And when I say get trapped, it cannot you cannot remove that. It can sometimes go back to the soil solution, but it's uncommon. It's difficult, and there's more need for research on this area. This is not a simple fraction to measure. It's not something you can send to a commercial lab and they're going to do it for you. And perhaps this is one of the reasons why we don't have a lot of literature related to this topic. Another thing that can happen to your ammonium is it can be lost via leaching, runoff, and erosion. Now, when you hear ammonium leaching, a lot of, especially those soil scientists, they're going to be like, what? It's not something you expect in a soil that has high CEC, but it can definitely happen in soils with lower CEC, such as Brazilian soils, oxysoils, and sandy soils. Um, Another pathway is your ammonium is going to be eaten by microbes. The microbes see that filet mignon there, and they go and eat it. And then now it becomes soil organic nitrogen. It's immobilized by the soil organic into the soil organic matter. That's not bad. It's not ideal, but it can always go back. It can be remineralized to ammonium, and one day can be used by the plant. So it's not bad. Another pathway is nitrification. This ammonium can be reduced to nitrate. There are steps. And I'm not going to go in details. You need different bacteria, two different specifically bacteria, and you firstly convert it. My mouse is not quite working. You first convert it to nitrite and then nitrate, but it doesn't really matter. This process, I marked it in red specifically because nitrate is extremely mobile in soils, especially the soils here in the Midwest, the mollusols, the prairie soils. They don't have really capacity to retain nitrate, so you can easily lose nitrate by leaching. Nitrate can be returned to ammonium. Not worth mentioning here right now, but it can. That's why I put the marker there. It can also be eaten by microbes, can also be immobilized. However, microbes prefer ammonium than nitrate. So if you have both free there in your soil, ammonium and nitrate, the microbes are going to eat the ammonium first because you can easily convert it in protein, not nitrate. So it can be immobilized and mineralized, but in a lower proportion than what would happen with ammonium. And then, of course, leaching, runoff, and erosion, you can lose nitrates and contaminate water bodies. It's something that everyone talk about when they talk about nitrogen. And another way to lose your nitrogen is via denitrification. This happens when after a heavy rain, if your soil is anaerobic, you don't have oxygen in your soil. It's a process that is carried out by several different organisms and several different reactions, and you can form different nitrogen gases. I'm sorry. And then finally, we can finally talk about what we want when we apply that urea as a farmer or producer or gardener. I don't know, it depends on what you are. Our goal is for the plant to eat it, and just a small portion of that nitrogen that is applied ended up. NDFF stands for nitrogen derived from the fertilizer. It's usually low. It varies. It can be lower than 20. I've seen 13, 15% in real farms uh, using isotope techniques, but it can also go higher. It depends on the 4 R's of nitrogen and what rate you applied, when did you apply, and etc. And it's what we try to maximize to increase farmers' profitability, to make their money worth more, right? The higher this number goes, the better for the farmer and the better for the soil as well. And just to finish the cycle, remember this is just a very short version. I'm not including animals. I'm not including biological nitrogen fixation and so many other processes here. The nitrogen can go back, can be refixed via BNF, biological nitrogen fixation, or chemical fixation again, just to finish the cycle for you. So green arrows are good, yellow are neutral, red are bad. And just to finalize, most of these processes, they're carried out by organisms. So soil moisture and temperature are extremely important, and they impact directly the rate of these processes. Other soil parameters, such as soil pH, will also be very important for these reactions. One final comment I think I forgot to mention. I marked the conversion for nitrate, ammonium to nitrate as red, also because during this nitrification process, you're causing soil acidification. You release two protons, which are acidity for each molecule of ammonium that you convert to nitrate. So over time, although we have calcareous soils in great parts of South Dakota, it's possible that you're going to see, especially on the surface where this urea is applied most times, an acidic profile or a pH that is lower or more acidic than what you would found down there. And that can be bad for plants, and we're not going to go in this topic today. Then now here I'm going to talk a little bit about immobilization and mineralization. And these depend on CN ratio. So what is CN ratio? So CN ratio is, for example, I have a wheat straw or a corn stover, and then I go and I do a total analysis of carbon and nitrogen, and I see that for each 60 carbons I have in my corn stover, I only have one nitrogen, or for each nitrogen I have 60 carbons. And here for wheat straw, it's more like 80. And if you have something very, for example, spruce, sawdust, or hardwood sawdust, we're talking about really high CN ratios. And then eventually, if you have more green stuff, the CN ratios start to drop. And if you have like a very green material, these are legumes that fix nitrogen, your CN ratio is even lower. And here's the CN, and these are just averages, they range, okay? Have it in mind. For example, the bacteria, here they're putting an average of 5, but it's something that can vary from 3 to 10, and sometimes it vary even more. These are just averages. So your organisms, they're gonna be on average like 1 to 10, 1 to 8 is a good average for all of them together. And for the soils, your A horizon usually have a slightly higher CN ratio than your B horizon. And potential reasons are you have more carbon on the surface. And in the B horizon of these soils, you can have nitrogen fixed in clay minerals like we talked about. So it's some potential reasons for why the B horizon usually have a lower CN ratio than the A. And what I want you to understand here is if your CN ratio is smaller than 1 to 20, you're going to most likely promote mineralization. Mineralization means I'm going to be converting my organic compounds, such as protein, amino acid, amino sugar, etc., into either ammonium or nitrate, which forms that can be taken up by plants. And immobilization is just the reverse, ammonium and nitrate to these organic compounds. I want you to understand that synthetic nitrogen fertilizers promote mineralization of soil carbon organic or of soil organic carbon and nitrogen because you're adding straight nitrogen. So if let's say my soil has a bunch of residues like corn stover, and then I add a nitrogen fertilizer, I'm going to be promoting mineralization of this corn stover, right? Because I'm reducing the CN ratio. And finally, mentioning that other nutrients are also important to finalize the equation in terms of mineralization and mobilization. And other nutrients would be, for example, phosphorus and sulfur. Now, the the main talk or the main topic that I brought to talk with you here today is the effect of synthetic nitrogen fertilizers on soil organic carbon and nitrogen. I decided to bring these two publications. I know both. I know the two first authors here, Saeed Khan, Dr. Saeed Khan, and Dr. Richard Mulvaney. Richard Mulvaney was my former advisor at the University of Illinois. I talked to him. He said, "Fine, I want you to spread the word about this topic." I think it fits very well the presentation. You're going to see the results, and don't even worry about carbon sequestration. What the message I want to talk about here today is: are we degrading our soils or not? Is this bad for the farmer? Yes or no, and why and how? So these two papers, both, they use the Morrow plots. The Morrow plots is the oldest experiment established here in the U.S. It was established in 1876, and it's. I work in a building that is just right in front of it. It's small. If you convert your scale here, you're gonna see that we're talking like maybe a little bit bigger than a football stadium, football field actually. And the subplots are maybe what 10 yards by 10 yards, 30 feet by 30 feet. They're small, and but they're very old, and there's data for over 50 years using the same practices here. And in their paper they're using the shaded plots. There's three different rotations: continuous corn, from since 1876 to present. There's a corn oat and alfalfa hay since 1876 to present. And in plot four, it used to be a corn oats, and then it switched over to corn soybean in 1967 because corn soybean started to gain more importance, and they decided to do this change. So we have data here. They're comparing data from 1955 to 2005, and there's basically three different treatments. One is unamended. Unamended means no nitrogen fertilizers were added. It doesn't matter if it's organic or if it's inorganic, nothing, zero. They're just draining the soil. On the second treatment, they're applying NPK using the Illinois University recommendations. So, which still is yield-based. And if you make a conversion here, it's going to be around 200 pounds of nitrogen per acre applied. And in terms of P and K, they did not change. They just followed the recommendations as well. In the high treatment, they're going to be applying 300 pounds per acre of nitrogen. So it's 100% more. No, wait, 50% more, right? A 50% increase in rate. And then these here were not used, so we're not going to talk about them. The manured site. Although I wish I had seen the data to make a comparison as well, but we don't have it. So here's the results, and the results are very, very interesting in my opinion. So as you can see, here's the corn rotation, continuous corn rotation. The corn soybean rotation, at least since 1966, if I'm not mistaken. And here's the corn hay, alfalfa hay. In all cases, a high dose of NPK and of N increased or caused a net loss of soil organic carbon. The most dramatic case was actually this one. If you make the calculations, I decided to use the corn soybean one because it's the most common rotation for row cropping here in the Midwest, if I'm not mistaken. And just look, in 51 years, they lost 4.8 grams on the very surface, 0 to 15 centimeters. If you convert it to inches, you're talking 0 to 6, 6 to 12, up to 18 inches. So I just added the 4.8 plus 4 plus 3.7, 12 and a half grams of organic carbon uh, lost per kilogram of soil. And this is just some conversions using the metric system. We don't have to go over them. uh, I did have some assumptions. I'm assuming the soil density is equals to 1.0. Soil density in Illinois is usually slightly lower than here in South Dakota, but I'm probably slightly wrong here. It could be 0.9, it could be 1.1, and that's going to have some effect, but not much on the final result. And I'm also assuming that soil organic matter contains 58% carbon, which also is just an assumption. We know that it can vary. It could be 55, it could be 60, but 56 is a number that it has been used in literature. It can be used to convert. After making all these calculations that I'm not going to go over them with you unless you want me to, you're going to see that in 51 years, they lost 49 tons of soil organic matter per acre. And that's a lot of soil organic matter. So for those who were here yesterday during Anthony's presentation, you're going to remember Anthony mentioning that if you want your soil to be healthy and if you want to increase if you want to have a regenerative system, you need to increase soil organic carbon or soil organic matter and not decrease. And this is exactly the reverse what these treatments are causing for the moroplots. And what does it mean? What means losing this 49 tons of soil organic matter per acre in 50 years? Well, you lose your soil organic matter, you're going to directly decrease your water retention and infiltration. That means you're going to be more prone to, for example, you have problems during a drought, or if you have a heavy rain, a heavy precipitation event, you're more prone for waterlogging or flooding because your infiltration is reduced. You're reducing your aggregate stability and structure. That means your roots are not going to grow as well as they used to be. Therefore, your plants are not going to take up as much as much nutrients. They're not going to grow as much. Soil organic matter retains nutrients. As I mentioned, they have a lot of CEC. So you're not going to be retaining these nutrients as much, and you're going to be also losing some of them, of course. You're going to be losing buffering capacity. This means resilience. Organic matter is well known for its buffering capacity, its ability to maintain, for the soil to resist to drastic situations. And at the end of the day, with lower organic matter, you're going to expect a lower microbial activity. It's what research data shows. If you have a very sandy soil with low organic matter, you're going to have lower microbial activity than if you compare then when you compare it with a soil with higher soil organic matter. And here's some indirect implications. And again, I'm going to cite Anthony. Yesterday, he showed a graph where it's clear as crystal. I don't know if that graph was from data just from South Dakota, but it's the same trend in Illinois. The soils that yield the best are the soils with more organic matter. In Illinois, they use a lot of alphisols and mollisols. Usually the alphisols have less organic matter and less CEC, and what you see is they usually yield less than the mollisols. It's very common to see that. So you have a chance to, in the long term, reduce your yield, and therefore, you can reduce your land value, right? If you can't produce as much, that land's not going to value. It's not going to be so good. And on top of that, you're going to be increasing by reducing organic matter, increasing your risk of erosion, which is terrible for our soils. I wasn't here, and probably none of us was during the Dust Bowl, but the pictures are out there, and it can be drastic, the problems caused by soil erosion. And then the story here is when Mulvaney tried to publish this this is over 15 years ago, right? This is 2007, if I'm not mistaken, or 2009. He heard about a lot of background from the reviewers. Oh, but this is just, you're using a football size field to collect data and tell that soil, that nitrogen fertilizer are burning soil organic matter. That's not right. You can't do that. So they did a step back, went back to the literature, and did a huge review on all sorts of experiments, including South Dakota. This is, if I'm not mistaken, I'm not sure if it was, I think it was Schumacher's student. Maybe Anthony Bly is going to be aware of his name, but Schumacher, I know that he worked with Schumacher here in South Dakota, and all of our neighbors that produce more corn and beans, let's say like that, like North Dakota, Nebraska, Minnesota, Iowa. Of course, I'm not mentioning Montana and Wyoming here. They did not have data for those two states specifically. Some other countries too. And what you can see is they're comparing the effect of high nitrogen fertilization rates or nitrogen fertilization rates on soil organic carbon. And the results are clear. And for these 43 different locations, I don't know how many locations I had in total, but you had a reduction in 43 of them and an increase in 4 of them. So more than 90% of the times, you're going to have a decrease in organic carbon, saving some exceptions, of course. So this is not only results that happened at the Mora plots. This can be extrapolated. This was found all over the central north USA and other countries too. And then here are the results for nitrogen. I decided to, I'm not going to talk much about the nitrogen part, but it's the same trend. The only, this is the only observation where there was a net increase in soil organic nitrogen after the 51 years. And for all treatments, even the regular NPK, there was a decrease in soil organic nitrogen. So that's kind of interesting, right? It's counterintuitive that I'm going to be applying more nitrogen, and at the end of the day, I'm going to be losing nitrogen. In my brain, it doesn't make sense. Well, it does when you understand the biogeochemical processes behind the story. But if you just think straightforward, it doesn't make much sense. But that's what the data shows, and in most cases, that the differences are significant. Okay, and so this is just a to further reaffirm. Here they're measuring total nitrogen, so it's all different forms of nitrogen in the soil. And here they're just focusing on the nitrogen that is potentially mineralizable. We're talking about forms of nitrogen less recalcitrant. They're like smaller compounds such as amino sugars, amino acids, proteins, things like that, that would more easily be decomposed. And they estimated this fraction using the Illinois soil test. And as you can see, there is a drastic decrease in all cases for all rotations. This one not as much as the other ones, apparently. I don't have stats here, but apparently there was a significant difference in all cases in terms of PNM, potentially mineralizable nitrogen in all rotations after 50 years. It's a further confirmation that you're losing organic nitrogen, and you're losing the potential of your soil to provide nitrogen to the plants, remembering that the soil is the main source of nitrogen for plants. Okay, I'm sorry. So how come excessive N fertilization rates reduce soil organic carbon, soil organic nitrogen? Well, high amounts of inorganic N reduce CN ratio and stimulate carbon and nitrogen utilization by microorganisms. That's the simple answer. I recommend going and reading the papers. They give more reasons, and they go in more details and more examples and how different management practices would be involved, and etc. But this is just a very, 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 very basic answer if you're thinking just about the nitrogen fertilizers. Now, do the nitrogen for are high doses of synthetic and bad only for soil organic nitrogen, soil organic matter? Well, this is data obtained in a paper that it's under review on a research that I did during my when I was in Illinois, right before moving here to South Dakota. It's not yet published. I shouldn't be putting it here, but I am. I'm not showing stats. I don't think stats are needed here. I didn't do stats yet, but the numbers tell the story. Maybe I would need stats to compare the gray and blue, but I don't need stats to compare the gray, blue, and yellow. The yellow is always significantly lower, and the yellow here is a conventional ag. It's a conventional farm applying 200 pounds per acre of UAN. They do pre-planting plus side dress every year. They have been doing it for the past 10 years. And this is a side by side, each farm. And here I have organic farms. They're both university farms, 1 mile apart. And this is a certified organic farm. They don't apply synthetic nitrogen fertilizers. And look at the potential of these soils, very same soil series. We made the characterization, everything the same. Look at the potential in nitrogen fixation. We're significantly so VE here, it's V emergency. This is we did our sampling 4 times during the growing season at V emergency and V9 for corn, V tasso and R6. And in all cases, there was, I'm not going to call significant because I don't have stats, but I know they are significantly lower N2 fixation by free living nitrogen organisms in conventional ag than in organic ag. Take home message. Well, I decided to bring this again because this is not a message I learned during my undergrad and most of my grad school. I just learned it later on, and I think it's extremely important because uh, I've been working with nitrogen for a while, including nitrogen fertilizers. And whenever I hear a nitrogen talk, we always hear environmental pollution, water and air. And sure, these are important, especially nitrate pollution. That's really bad stuff. Clarence talked about eutrophication. You need both nitrogen and phosphorus together, and you have eutrophication. That's terrible for water bodies. But I'd like to bring the fact that excessive nitrogen fertilizers also inhibit biological nitrogen fixation. So you're reducing the capacity of your soil to produce more nitrogen, naturally produce more nitrogen for you. And in the long term, you're going to be burning your organic carbon and your organic nitrogen. And that's going to, as we talked about before, reduce your potential yield and lower your land value. So if I was a farmer, I would be more worried about this than this. But this is, I would be definitely, if I'm thinking about future generations, I'd be definitely thinking about this when I talk about nitrogen fertilizers. And what are potential solutions? Well, I just have 2 minutes, so I'm going to be fast here. Well, for those who want to stay in conventional ag, 4 hours of nitrogen, improving the rate, the timing, how you apply it, what source you use. It's a great way to do it. There's a lot of people here in South Dakota at SDSU, such as Jason Clark, Anthony, folks out in the West as well doing research on it, and they're doing a great job. They're doing great here in South Dakota. In my view, South Dakota is doing way better than what we do, what is done in Illinois when it comes to nitrogen fertilization. I'd also like to mention variable application rate, especially based on soil and management practices. I believe that this is something that really has potential. And for those willing to do more drastic changes, I would mention diversifying your rotation, implementing no-till, very important for maintaining organic matter. Use of organic amendments such as compost for those who were here at Anthony's presentation yesterday. And lastly, but the least but not last but not least important, livestock integration. I also think it's extremely important. Here is just an example of the. I'm not going to have time to talk about it right now, but it's just like a variable rate based on the ISNT, which is a method to predict potentially mineralizable nitrogen. And the rates vary according to how much nitrogen can be potentially applied. And to wrap up, I'll take this extra minute to talk about this one. This is Sergei Winogradsky. He's the first microbial ecologist. He's responsible. This guy is responsible for discovering nitrification process, free living nitrogen fixation. He was the first to isolate Clostridium, which is an anaerobe nitrogen fixer. And he was also the one who proposed and proved the concepts of autotrophy and chemolithotrophy. So big name, extremely respected. And he wrote this when he was almost 90 years old. He's the kind of guy who worked until he died. One of those. There are several of them out there. A lot of farmers are like that. And he wrote that when he talked about the law, he's talking about the fact that mineral N inhibits biological nitrogen fixation. He says that when this law is confirmed, the farmers will one day, perhaps the farmer think over the fact that in pay for nitrogen fertilizer, he deprives his land of nitrogen, which nature gives him for nothing. A lot of scientists and folks are going to say that I'm crazy, and I am, and probably I am, but my dream is to see one day a system where we're applying the least amount of nitrogen possible, more focused on organic forms than synthetic forms, and maintaining profitability and increasing our soil health in a regenerative agricultural way. I'm not going to have time to talk about the projects I'm involved with. I'd just like to thank you. This is my contact information. I'm here at Raven in Brookings, the agronomy department. My office is right where I'm in right now. It's 213. I spend most of my time at the lab, 130 Soil Fertility. Here's the address. I'd be glad to take your questions.
Thank you, Vander.
Yeah, you bet.
Thank you. Yeah, we do have a couple questions.
Sure.
First, I'd just like to remind everyone, thank them for attending. These recordings will be on our SDSU Extension Crop Hour webpage. Later, you will be getting a survey. Please fill that out. It's important for us to learn how to do better in these sessions. And just want to thank you again for attending. So let's get to the questions. First question that I saw was about the Morrow plots. What's the tillage history of those plots? And I'm going to even add just what do you think the effects of tillage might have had on that data that you showed?
Definitely. I thought about the same thing when I was preparing the presentation. Mulvaney's paper does not specifically mention, but I did look out. Oh, here we are. I did take a further look in other papers, and at the Morrow plots, they're using conventional till. And we know that conventional till will promote carbon oxidation, or you're going to be promoting mineralization. You're going to be burning some organic matter, in short words. And during the past years, it has been conventional tillage, but in the past they used to do chisel plowing. So and more board plowing, chisel plowing. It just changed. I'm not exactly sure about the time frame, but unfortunately, they do not have, for example, a long-term no-till there at the Morrow plots. Here, when we look at the treatments, we're able to tell the difference, the effect of fertilizer only because, for example, this plot did not receive any fertilization. The tillage is the same for everyone, but the results are different according or not to how much nitrogen you apply. But yes, I definitely agree with you. Great question. I thought about the same, and I should have added it here. This is conventional tillage. Currently.
Oh, that's just fine, Vander. So there was any other questions? Want to make sure that everybody gets a chance to ask a question here. I think a very intriguing presentation. Lots of good information there. We think we're doing the right thing sometimes, and it's not necessarily the truth. So we have to think that through. I have a question on your last slide.
Sure.
Yeah, but I took a picture of it. Um,
Uh-huh. Oh man, I'm trying to find it here. 1 second.
so Dr.
Immediately find. Yep,
Winogradsky,
Sergey
was he a
Vinogradsky.
Russian or Polish descent?
Well,
You know,
uh,
doesn't matter. But
I actually know he's back. He's from Ukraine, but back then, oh,
oh,
QR code. He's from Ukraine, but back then, Russia, it was the Russian Empire. So Ukraine and Russia were the same
oh yeah,
territory. He was born
okay.
in.
There was a term down there.
Mm-hmm.
He was basically the discoverer of many things, concepts, and then
Yeah,
chemolithotrophy. Could you talk about chemolithotrophy?
sure. So some organisms they are going to use mineral compounds such as nitrogen and sulfur as final electron acceptor. What I'm trying to say is, for example, Vander as human, I'm a heterotroph. I use glucose to do the respiration and to make energy for my cells to work, to regenerate my ATPs. The chemiolithotrophs, they're going to use stuff like sulfur and other compounds as final electron acceptor. So that's the difference. They don't use carbon as final electron acceptor.
Very good. And you know, this is very you can tell by his quote there that his English. That's why I brought up the fact is he Russian or Polish or whatever, because his English is broken and we'd expect it to be. So, but it's very
Yep,
well said in his quote there, and I think we're finding that out today in a lot of different ways. So
yep.
maybe address the concepts of autotrophy just a little bit.
So the autotrophs, they're able to produce their own energy. So, for example, the plants are autotrophs, right? They don't need to eat carbon. They can just do photosynthesis and convert CO2 into sugars and then use them. This would be an example of autotrophs. Yeah,
Okay,
did I,
very good.
did I get
Just
it?
kind of want to highlight those two things for others to learn about there, Vander, and
Sure.
thought that it was kind of neat that you had that on there. Um, anyway, what's old is new again, right?
Yeah, what is old is new
Basically,
again.
new
It's
again.
fascinating. Like when I think about Hopkins and Joseph Hutton, what they were thinking about ag back then, the early 90s, it's very similar to what we're shifting towards right now. They were on target, and then we kind of drift away with the synthetic end fertilizations, and then we started to see bad results from excessive end fertilizations, contamination here and problems here and there, and now we're getting back on track apparently, which is good.
Which is good. Exactly. And it's
Yeah.
all about the carbon.
Yep. You bet. In my opinion as well. Yep.
Yeah. Even just ignoring the last few years of talk about climate smart commodities and greenhouse gases, even ignoring all of that, um,
Yep.
the carbon is so important for the soil. So,
Yep. Yeah. I
yeah,
try to avoid as much as I could, like talking about this greenhouse gas stuff, because I think that the effect on soils are more important for the farmers, and they should be aware of it.
very good. Very good. Well, I think we've
Hey,
exhausted
Vander,
our.
did you get a chance to
Go
see
ahead.
the question I put in the chat?
Oh, I didn't. I can't see the. I can't. Oh, now I can see what I'm seeing. I'm sorry.
Oh, there it
Um.
is. I'm sorry, Clarence.
That's all right. That's okay. I figured
Yeah.
we'd
Tillage history. Oh yes, the tillage
no
history.
though.
Oh,
So it's in the
no.
chat, Vander. That's the
Oh,
Q&A.
I'm
I'll
sorry.
read it to you, chat. Have you heard of the proposed project in South Dakota, commonly referred to as the green ammonia pathway? I
Oh
first heard
yeah,
of it late January. Yeah, the green ammonia pathway. Have you heard of
yeah.
that?
No, I didn't hear about this specifically project. It is indeed true that we can nowadays produce anhydrous ammonia using water and electricity, water hydrolysis. It's 100% renewable. For example, let's suppose I'm using air, electricity plus water. It's very green. However, to my knowledge, nowadays it is still less expensive to produce ammonia using fossil fuels such as natural gas than a renewable source. So it would be that kind of stuff. Are you willing to pay more for something that is greener? If yes, great. If not, it's like gas. It's like electric cars versus gas. Not a good example, but similar.
Clarence, would you like to review that OI project a little bit? I mean, I've heard of it too.
Yeah,
but
would you mind explaining to us?
Go ahead,
Sure.
Clarence.
Well, I kind of have just limited notes from the actual project itself or what they were proposing, but what they were kind of emphasizing is that ethanol plants, for example, have a lot of carbon dioxide emissions, and there's been a lot of talk about what do we do with those emissions, and I don't really want to get into that. But basically, the basic idea was to use that carbon dioxide as an ingredient source for anhydrous ammonia or urea or things of that nature. And what was really stuck out to me was that, South Dakota specifically is not very fossil fuel rich. We're kind of dependent on fossil fuels, which is kind of surprising considering we don't have any oil wells or any natural gas supplementation of any kind. So that was kind of my big take home message. And I haven't heard much about it other than that one particular time. It sounds like a pretty interesting potential fertilizer source for South Dakota farmers, but I was kind of curious if you wanted to touch on the bigger picture, maybe some of the things that you had discussed today.
Oh, that's cool.
Yeah, I can expand on that a little bit. They're looking to use wind power, electricity to produce the ammonia, and then further that combine the ammonia with CO2 to make urea.
Right.
And so very innovative thoughts there coming from Central South Dakota, and I hope they're successful, because that's leading to the right step, I think. So
Perfect.
Yeah,
Thanks,
I
guys.
definitely agree with what Anthony said, Clarence. And I wasn't aware of this specifically project here in South Dakota. And I think it's great, honestly. I think if we have ways to reduce carbon emissions and producing fertilizers from this carbon, it's even better. It's a win-win scenario. And that, along with the 4 R's of nitrogen and nutrient management practices and all the good stuff that folks are doing here in South Dakota State University, it's the way to go, especially for those who want to continue cash cropping more conventional style, I'd say.
Perfect.
Okay, I think we've covered it all, and thank you all for hanging on with us. I think you've appreciated the little extension of conversations, because you're not going anywhere, and so that's telling us a lot. And so thanks. Have a good year, good cropping season. Hope it starts raining soon, and we'll go from there. So with that, I think we'll call it an end. Thanks, Vander.
Thank you all. Have a good
Thanks,
one.
guys.
Yep. Thanks, Clarence.
Yep. Have a good one.
Bye-bye.
Thanks, Anthony.
Yep.